Hey guys, Dr. Ben Edwards here. Welcome to another episode of You're the Cure. Got a great show today, but a few announcements real quick before we jump into the show. First, if you need a little holiday inspiration, we have got something for you. Little ebook, Deck the Table Harvest Edition. The team did an amazing job of putting this thing together. I'm talking top quality excellence. Amazing food, amazing recipe, nutrient dense food, of course, but so beautiful in the table, the place settings, everything professionally done. Highly recommend this ebook. You'll get a 3-course menu, 6 different recipes, the full grocery list, the links to where you can get all the table decor, the place setting, the silverware, the everything. A lot of talent and giftings from the team came together here. Bella with her beautiful plating of the food and her computer work to put this into an ebook format. Of course, Nicole bringing the amazing recipes. My wife, Jamie, bringing the excellence of her decorating skills. Just a beautiful table that Jamie did. We had Logan take some amazing pictures. Heather's our project manager, just the whole team. Talk about putting on an example of excellence in the kingdom. So y'all check that out. I'll be speaking at a few different places in the new year. I want to give you all a heads up. January 22nd, that's a Thursday evening, 5:30 to 7 down in Midland. That's the Truth About Your Health, Part 2. Had a great time down there with Simply Midland and Heather Claybrook and the group down there. Don't know the location yet, but mark your calendar, January 22nd. Be back down there talking about the truth, how to walk in that truth. And then February 27th and 28th, Aurora, Colorado. That's just outside of Denver. That's with Morley Robbins and the Root Cause Protocol family. Ignite the Innate Healer is the name of that conference. You can go to therootcauseprotocol.com and get your tickets. Early bird pricing ends January 15th. That'll be a great conference, great lineup of speakers. One of my favorite conferences, invited to last year. It was awesome. Loved Amish country up there in Wooster, Ohio. Seeking Whole Health. They do a fantastic job. So many great vendors, great speakers, great food, just an amazing, beautiful atmosphere. Love that conference. Early bird pricing ends January 5th. And you honestly, guys, if you want to attend that one, you better jump on this quick. I think they just opened up sales last week or this week and sold out, sold half the tickets, I think, in the first few days. So that'll sell out quick. Go to seekingwholehealth.com for those tickets, April 17th and 18th. And then the following weekend, April 24th, 25th, I'll be in Waco, Texas, Homestead Heritage. Great community there, beautiful facilities. It's thehomesteadconference.com is where you can see all the lineup of speakers, amazing speakers on homesteading. So I'm honored to be going to Waco for the first time to speak at this conference. And then looking a little more into the future, August 14th and 15th, Need Your Cure. Love the Need the Cure family over there in Oxford. Honored to be going back there to that conference in August. And, I wanted to tell you about all these conferences, guys, not only to support these folks, but and encourage you to go, especially if any of these are in your area. But even if you're not in any of these areas, I know that Seeking Whole Health, people come from all over the country to attend that conference. But I wanted to highlight these folks because this is an example of people who love the truth, who love you, who love teaching and supporting and walking with you on your journey into wholeness and health. I was at CHD, Children's Health Defense, this past weekend in Austin, and it was just great being around so many like-minded folks who love each other, love the truth, love learning the truth about how to walk in health. Today's show is actually kind of a little depressing. We have Dr. Jessica Rose coming up here in a minute, and she's going to bring the latest research, literally hot off the presses. Yesterday, or the day before we recorded this interview, she was in the lab discovering things about the COVID-19 injections. And kind of don't want to talk about that anymore. But on the one hand, I think it's important to remind ourselves um, about the truth about that gene therapy shot and what's being discovered even now, 5 years later, and what our health authorities are not doing anything about. So you don't get too sad during this interview. There is, of course, always hope, but I want to highlight these conferences as places you could go to get encouraged and to just know and to see for yourself that there's lots of people out there who are trying to look out for you and bring you the truth on healing. So I encourage you to check those out. And then just real quick, one more little thing I'd like to say. I was awarded the Defender of the Year by Children's Health Defense, and just want to say thank you to them for sure. But the reason I'm bringing this up is I want to publicly recognize Dr. Richard Bartlett and Tina Siemens and all of the helpers that Tina Siemens brought to Gaines County to help me during that outbreak of the measles. And of course, my family and team here at Veritas in Lubbock, the clinic just continues to operate and the wellness company operates because of the amazing team that I have to support. So it takes a full family, the whole body working together. And I know I'm the leader, the CEO, and there's a responsibility I have for sure to steward the team. But it's a total team effort. I know my role that I play, but I can't play all the other roles. Shouldn't play the other roles. Can't play the other roles. All the amazing and beautiful, true identities and gifts and talents that the whole team brings. So, just really wanted to recognize these other people. It's never one person when there's an award. It's never the one person that you know really deserves that. So just want to, if I could replicate that award and give it to the whole team, I would. Okay. I think that's it on the announcements. Let's jump in to today's show with Dr. Jessica Rose. All right, everybody, welcome to the show today. Got a repeat guest, Dr. Jessica Rose. I'll bring her on just a second. Just want a little fair warning. We're shifting gears a little bit this week. I was just at a conference down in Austin, Children's Health Defense. A lot of amazing people there, amazing speakers, an amazing audience, and just an amazing organization. And some of those speakers kind of brought me up to speed on some of the newest research regarding vaccines and mRNA in particular, and COVID-19 related issues. So, if you're new to the program, we were deep diving COVID-19 stuff back in the day, and you can go on the website, veritaswellnessmember.com. You can click on pod resources, then podcast, and there's a topic menu, and you can hit COVID-19, and there are 25 different podcasts. And we've talked with a lot of the experts, including Dr. Jessica Rose, who will be on today. And if some of the stuff we talk about today is a little shocking to you, well, welcome to the show. It's been a shocking few years. And today's show's designed to educate you, not scare you. It's designed to inform you so you can make some informed decisions and educate your family and your coworkers and your friends. So let's dive right in. Dr. Jessica Rose, welcome to the show.
Hi, thanks for having me back. Can't
You're
believe it's
welcome.
been 8 years.
I can't either. And I was trying to think, and really, I should have kept a diary all through 2020, 21, 22. Such an amazing time, and not always in good ways, but in good ways also. Somehow, a lot of these scientists and physicians connected, and I don't know how we all connected, but we all found each other through social media or through colleagues that were like-minded. We ended up on group text together. We ended up on different social media platforms where we weren't kicked off, and we found each other at conferences. And anyways, Dr. Rose is one of those folks. And really, Jessica, I thought we'd start with maybe you given some of that background. What were you doing pre-2020? What happened to get you so interested in researching COVID-related things, mRNA-related things, as opposed to some of your friends or colleagues or former schoolmates who just stuck to the conventional mainstream narrative?
I was chugging along doing academic research. Just finished up my last postdoc in protein biology and biochemistry at the end of 2019. So that was right before they declared this pandemic. And yeah, I study pathogens, viruses and bacteria using mathematical modeling. So I was well poised to be commenting about what was going on when this all started, when they started throwing around words like zoonotic pathogen, and it became pretty clear pretty fast that that's not what we were dealing with. We were dealing with new therapeutics, gene-based therapeutics that they were going to be trialing, I suppose, although they didn't really trial them. They just put them in everyone as an experiment, and a system of control, which you know we were all subjected to. People haven't recovered from it all. I'm hearing a lot these days that this is old news. Why are you talking about this now still? But it's such not old news for so many reasons. The most annoying one being that there's been no accountability for what's happened. And even yesterday and the day before, I spent 2 days in Kevin McKernan's lab, checking for DNA in the vials, in the COVID vials, both Pfizer and Moderna, and we found tons of it using 3 different methods. So this is not something that's yesterday's news, from that point of view, because DNA, first of all, it has no place in an mRNA product, as they pushed, and it has physiological and clinical implications, because there's so much of it, and it's introduced to the form or in the presence of a lipid nanoparticle, which is, it's a very efficient way to get genetic material into cells. A lot of people are wondering, well, what is that going to mean for us, like for the people who got injected with this crap? And I mean, the one word answer is cancer, but we can talk more about that after. Um, yeah, I mean, it's like an accident. You can't look away once you look at it, and because I have the background that I do. I'm skilled in data analysis, so as well. So I started combing VAERS for adverse events, and the safety signals started popping up, like completely. They were so anomalous. I mean, it was like a sore thumb sticking out, and that happened in January 2021. It was immediate. Yeah, still hasn't been addressed by the owners of the data.
Yeah. So, guys, for new listeners, VAERS, Vaccine Adverse Event Reporting System, which is vastly underutilized by clinicians and general public. Most didn't even know about it. Probably more do now because it has been in the news a little bit. But that's what Dr. Rose is referring to with VAERS. So this abnormal signal, suddenly there were all kinds of reports coming out of adverse events and deaths related to the COVID injection.
And by all reports, we're talking about like over 1.6 million reports. And just for context, that's just in the COVID shot context. And for the last 30 years, since VAERS was incepted, for all vaccines combined, the total number of adverse events reported per year was about 39,000. So there's zero comparison there.
Yeah. So as you started to see that initial safety signal, where did your mind go as far as causality? And then what has the research showed as far as what could be causing that safety concern?
Well, it depends what you're talking about. I mean, what's become evident is whatever somebody might be predisposed to, and that includes people who are older who might be near death. It's very, very clear, just from an outsider's point of view, that the shots are pushing everyone toward whatever that predisposition is. So let's just say you have an autoimmune condition. that's been suppressed because, I don't know, you're taking good care of yourself and you're eating the right food, getting enough exercise. If you get one or two of these shots, it seems to, without fail, cause a reemergence of that autoimmune condition. And that's based on immune dysregulation and hyperinflammation, which is, it's obviously going to be induced by overproduction of a foreign protein by your own cells, overactivation of your immune system, destruction of your red blood cells, both because of the LNPs and because of spike. Yeah, there are actually many mechanisms of action here, but the bottom line is that the immune system is just being completely dysregulated in every possible way you can imagine. Um, it's just creating imbalance. It's the easiest way for me to put it, and it's irrefutable that it's doing that. So
Yeah.
from myocarditis to I don't know, autoimmune or to like demyelinating conditions, let's say. It's across the board. And as far as causation goes, there are ways to assess that, and that's actually what VAERS is for. It's a pharmacovigilance database, which the owners of the data, which are CDC and FDA, are meant to use to ensure that once you go from the phase 3 clinical trial, which only has, tens of thousands of people, to a much larger demographic, and in this case we went to like billions of people, that you don't see emergent safety signals like myocarditis. So if you see a cluster of adverse events being reported, or an emergent signal that's kind of weird, like male breast cancer, you do, as par for the course, a causality assessment using a variety of methods like proportional reporting ratio, Bayesian analysis, or Bradford Hill criteria. And I've done all this, and there's no room for argument here. I mean, the shots are causing a lot of these adverse events. When someone is injected with a product and they go into anaphylactic shock, for example, within 15 minutes, nobody is going to refute that they went into anaphylactic shock because they were injected with something. Like no one's going to argue with that. So we're seeing a lot of that. It's an analogy, but like one of the criteria that you have to satisfy in this Bradford Hill criterion thing that I mentioned is temporality. And what that means is that, first of all, one thing has to come after the other. You get injected and then you have the emergent adverse event. And then the shorter the time frame between those two things, the higher the probability that there's a causal relationship. So this is all stuff that's, it's based on common sense. It's based on what we've done in the past. And for some reason, in the context of this nucleoside modified mRNA LNP platform, everyone seems to have gotten amnesia pertaining to what we've always done. And what we've always done is supposed to be in the interest of so-called public health. But I actually don't believe there is such a thing anymore.
Well, there's plenty of things going on to kind of make someone come to that conclusion, I would say, including this. I just read this article the other day out of Japan. And basically a Freedom of Information Act was, a FOIA request was submitted. So 21 million Japanese citizens, their vaccination records were released and their health records, and the conclusion. And this is Professor, I'll butcher the name, but Murakami of Tokyo Science University, a well-respected professor. And based off this data, 21 million Japanese citizens data, 600,000 to 610,000 Japanese citizens most likely died directly due to the COVID-19 mRNA injection. and this death surge was within 90 to 120 days post-injection. He saw a cumulative toxicity effect also. That just came out last week. Freedom of information request. Similar freedom of information requests have been submitted here in the United States. I know the Pfizer document showed it, and don't quote me exactly, but something like 1,200, 1,300 deaths. I think it was the first 90 days reported to Pfizer. Of course, they wanted to not disclose that. I think that may have been a court order. They had to disclose that. So, yeah, when the numbers like this, and it takes a Freedom of Information Act or a court order of some sort to get these kind of statistics, where the public health agency should be leading the charge on this investigation and finding these answers and then doing something about it. Yeah, it's hard not to conclude where's public health on this matter, but all the way up to the very top, these congressional hearings a few weeks ago with Robert F. Kennedy Jr. and the former director of CDC, and just listen to these folks. I mean, it's so partisan, obviously. Politics, can't hardly stand it. So yeah, I understand where you're coming from with that conclusion on where's public health at. Could maybe you talk a little bit more about what you presented at Children's Health Defense this weekend and some of this, maybe for the layman who's not educated on mRNA, nanolipid particles, and the DNA contamination you just talked about, but talk a little more about what specifically is the issue. Is this getting incorporated into our DNA? Is it being broken down? Is it being biodistributed? You know, if someone got an injection 3 years ago, is it still having an effect today on their DNA? That kind of thing.
So from back to front, we don't know. It's very variable. That's a certainty that we verified yesterday, actually, in the lab. But so basically, what I talked about at the CHD event, which was wildly successful, by the way, there were like over 1,000 people there. You couldn't walk a foot. Well, I couldn't walk a foot.
Yeah, jam-packed, sold out. I don't know how many virtual tickets they sold to. People are very interested in finding like-minded folks and finding people speaking the truth that they can trust.
People want answers, and
Yeah.
that's what I'm here for. So in
Yep.
any way I can help, I'm going to. But me and two colleagues, Kevin McKernan and David Speaker, recently published a paper in Autoimmunity on our findings, which are, irrefutable evidence that there's DNA in the Moderna and Pfizer vials. And just to refresh everyone's memory, we were explicitly told that there's not going to be any problem with, DNA integration or genomic instability, which is when your genome becomes unstable, and that's a bad thing, because these products were mRNA based. Now, granted, they're nucleoside modified mRNA based, which means that it's very stable mRNA coding material. So they did that on purpose because they don't want the coding material that your cells are going to translate into spike protein to be degraded quickly, because mRNA is, it's very degradation sensitive. So they modified it such that it wouldn't get eaten up by, your host cell enzymes. So during production, first of all, I have to run everyone through what they did, which is also part of our paper and what I talked about at CHD on stage. There's two processes for making this nucleoside modified mRNA that they packaged in these lipid nanoparticles, which are basically just little fat bubbles that carry whatever genetic material you want. There's process 1 and process 2. So as part of the clinical trials, that's like the test stuff, they use the process 1. And what that means, the difference is that the DNA coding template that they used was produced using PCR, polymerase chain reaction. That subsequently went through an in vitro transcription reaction to produce this nucleoside modified mRNA, which was, we hope, cleaned and then wrapped in the LNPs and injected into people as part of the clinical trial. And the data was collected. Any adverse events that arose as part of that were reported. And as you already stated, there were a lot of adverse events that occurred in the context of those clinical trials. Even though they used that different product, the data is still crap. That's very important to bear in mind. But here's an even worse thing that they did. So when they needed to inject billions of people with this stuff, they needed to upscale production. They wanted to save cost. They wanted to make it fast because, we were in an emergency and we needed to get everyone injected right away. So what they did was they switched to process 2, which involved something called an E. coli plasmid system. So instead of using PCR to amplify your DNA, they exploited the doubling power of E. coli bacteria that were transformed with these DNA plasmids, which are little circular pieces of DNA that carry the spike gene, to make tons and tons and tons of this spike DNA.
Because E. coli replicates every 20 minutes, I think you said.
Yeah,
right?
it doubles every 20 minutes. So all you have to do is give them a little bit of, sugar and warmth, carbon dioxide, and shake them up overnight. And when they double, most of them are going to bring along this plasmid, this little circular piece of DNA that carries the spike gene. So you're going to effectively overnight end up with a ton of your spike DNA. And then what you're going to do is you're going to break up your cells, take that DNA, out of circular form into a linear form, which they also screwed up, and then do an in vitro transcription reaction to convert that to mRNA. And in the case of what they were doing during production is they created modified mRNA by introducing N1-methyl pseudouridines instead of uridines, or to replace that base. So then what they were supposed to do, and this is a very important step, at the end of the production process is clean out all the junk in the Eppendorfs or whatever they were doing the reactions in. That means all the DNA from the plasmids, you don't want that in there. And lipopolysaccharide, which is it is the component of the cell membrane or the cell wall of the bacteria. Because if you leave that behind, if you don't clean out that and you inject it into someone, they'll probably go into anaphylactic shock. It's really toxic. So this last cleanup step is really, really, really important. And the way that you get rid of DNA is you just use an enzyme called DNase. In their case, they use DNase 1, which is a specific enzyme that chews up DNA, and then you're supposed to filter it out. You're supposed to be left with only nucleoside modified mRNA because this is supposed to be an mRNA product like they promised. But something went horribly wrong from many facets at that endpoint, because we are finding, and this finding has been reproduced in labs all over the world. We reproduced it yesterday and the day before in Kevin's lab. We tested 8 new vials. There's tons of DNA in the vials. And you can only assume that since there's tons of DNA, and I mean tons, that there's also probably lipopolysaccharide. Now, that's an assumption, but what this is definitively leading us to understand is that their enzymes didn't work, or they did, but ineffectively, and the cleanup process wasn't done properly. And so all this stuff that's not supposed to be wrapped up in the lipid nanoparticles, which are also toxic, by the way, that's not supposed to be in there, is in there. So billions of people got injected with this crap. And by the way, this process 2 methodology, the synthesis methodology, they only tested this end product on about 250 people. Not 250,000, 250. One lot. That's not a clinical trial. it's a new product. So they needed to do brand new trials for this new product. It's deplorable what they did. It's deplorable. There's no excuse for it. Even if we were actually in a real emergency, there's no excuse for this. You don't cut corners like this when you're talking about an experimental gene-based therapy. No, you don't. So what we're left with are billions of people who've been multiply injected with this crap, and every single vial that's been tested, not just by us, but all over the world, contains excessive levels of DNA. And by excessive, I mean levels that exceed EMA standards. That's the European Medicines Agency. These are the people who, and the FDA, they're the ones who assign, the so-called safe levels of DNA that you can inject into a person and not have them, drop dead or whatever. But it's really important to understand that those levels are already way too high because they're based on naked DNA. That's not lipid nanoparticle encapsulated DNA. That's naked DNA. It's just DNA introduced to the body, and it has a 10-minute half-life. But that's not what we're talking about here. We're talking about something completely different. These little fat bubbles that Moderna and Pfizer designed are protective capsules, and you're talking about the protective encapsulation of already very stable molecules by design. And this DNA, which we, I won't get too technical, but we're hypothesizing, and we're pretty damn sure that the reason why this DNA got in is because it hybridized to the RNA molecules, and that there's only one specific DNase, an enzyme, that could possibly, destroy and or remove that. And they either knew or they didn't know about it. But the thing is, there's no excuse for them not to have known about this, since they absolutely knew. We have the documents to prove this back in March 2021 that there was a DNA issue. So I know that was long-winded, and I hope it was
No.
understandable, but
Yeah, that's great. But talk a little bit about what's that matter? What is DNA, foreign DNA in the body, especially when it's stable in a nanolipid particle? What are the implications of that? Where does that DNA go? What does it do? And what are the clinical implications of that?
So the modus operandi of these lipid nanoparticles, first of all, is to get everywhere in the body. I don't know. I don't even understand why they would have said that stays at the injection site. This is another blatant lie that we were told, because these lipid nanoparticles, they're like little 100 nanometer across fat bubbles. They're designed to go everywhere in the body via the bloodstream. That's the whole point of an intramuscular injection. So everywhere those trillions of molecules are going, they can potentially transfect whatever cell. We don't know which cells. We don't know how many cells. And that's why there's so much variability. And then what happens is whatever genetic material is inside those lipid nanoparticles, or even on the surface, because they could be adsorbed, i.e. RNA, modified RNA, or DNA, or whatever other, top problematic factors there are, gets dumped into the cytosol of the cell. And if you simply, and this is all documented, this is in the literature, it's not a secret, it's not something we discovered. If you simply introduce foreign DNA fragments into the cytosol of a cell, this has nothing to do with integration. You can induce cancer pathways via inflammatory pathways like the cGAS-STING pathway. So there's not even a need for integration to occur, which most likely is happening, but you don't need it to induce cancer pathways and highly inflammatory pathways. That's what the immune system will do. I mean, if it sees foreign DNA, it's not just going to sit there, it's going to respond. So cancer is like, everyone is hearing about somebody having some weird cancer, or young people getting cancer, or aggressive cancers are two words I'm hearing a lot, rare cancers, ever since 2021. And so it begs the question, is this DNA impurity issue leading the way or spearheading all of this cancer, you know, um, the clinical cancer reports? I imagine it is. But if we have integration as well of this, of whatever DNA fragments happen to be getting into people's cells, and then potentially into the nucleus of their cells, that's horribly problematic, especially if we talk about germline integration. Those are your, your stem cells, or your sperm cells, or your all the reproductive cells. This is horribly problematic, because then we're talking about generational problems.
Yeah. I just read a study the other day, or a case report. A 31-year-old, stage 4 bladder cancer, just that all by itself, very unusual. But
Yeah,
then they did a genomic study on that, biopsy, and a 20 nucleotide segment from the Pfizer mRNA.
yep,
It's exactly what you're talking about.
it is indeed, and that kind of is proof positive that integration is occurring. This is why we need to talk more and more about this, which is why I'm incessantly talking about it now, because we're years into this, and so much of the damage has probably been done, and a lot of people aren't connecting the shots to whatever cancer they might be dealing with right now. I mean, it's unless you're kind of in this and looking at it, it might not occur to you that, oh my god, it was because of those shots that I have bladder cancer. So we need to make these connections and stop allowing the suppression, and design assays, like tests, to verify what our hypotheses are, which is that this crap is integrating. And if so, we have to define the mechanisms of action, and we need to find ways to help people. We need to find ways to purge this deadly spike protein from people. Um, and we absolutely need to stop injecting people with this crap, or any crap designed based on this platform. Because it's the platform that's problematic, right? You can't really do this nucleoside modified mRNA thing without the LNPs. Um, so we need to, yeah, a moratorium needs to be called for sure.
Yeah. Here's another study, South Korea. This is pretty recent. I was looking at the other day. COVID-19 vaccination could be associated with an increased risk of 6 specific cancer types, including thyroid cancer, gastric cancer, colorectal, lung, breast, and prostate cancers. And there's plenty of other studies. That was just a most recent one that I saw. Moratorium. I've heard some other folks like Dr. Peter McCullough call for that too. I think I don't hear any health agency calling for that, or anyone in a place of authority calling for that. And in fact, I think I've heard that they're looking to potentially use this platform, the mRNA platform, lipid nanoparticles, on the childhood vaccine schedule. I just heard that actually at the conference. Haven't looked into that. Have you seen anything, reports on that same topic?
Yeah, well, it's already on it, but it's hopefully being removed. I thought it was. But the more ironic thing is that there's, for quite a while now, there's a cancer therapy based on this platform being designed. So that's all par, and you know it's all par for the course, right? You induce the cancers, and then you come up with the solution to solve it. It's a money making scheme, if you ask me. Um, One of the things I forgot to mention is that one of the DNA fragments that we're looking for and have detected in copious amounts is something called the SV40 promoter. Oh, here's my cat. Promoter.
Hey, I remember that cat last time I interviewed you, or maybe it was a different cat.
No, it's the same cat.
Yeah, little kitty, kitty.
Thing.
My wife's gonna love this podcast.
There's your butt. Yep. Show everyone your butt nuggets. Come on.
Nuggets? Is that it? That his name?
No, those are
Oh,
never mind.
yeah, I'm tracking.
Sandy, come on.
Oh, cat lady. I didn't know you're a cat lady.
There you go.
Which, uh,
Okay.
a little side note, and then we'll get back to what you were just about to say.
Yeah.
It was
Make
just
sure make sure that stays in the video. Everyone loves him.
oh, yeah, there's no way we'll edit out a cat. The public doesn't know I'm kind of a cat man. Just kidding. I said that to make my wife really laugh. She never listens to the podcast, I don't think, but I'm gonna tell her to listen to this one so she can see the cat.
Please, my
Um,
so
SV40, I think you were saying.
yeah, we found a lot of this, and SV40 promoter enhancer is a component of the simian virus 40, which is this virus that was polluting the polio vaccines way back when, which is it's an oncovirus. I mean, it causes cancer in us and many mammal species. This is definitively known. So this component part of the SV40 virus, the promoter enhancer, is used as a gene therapy tool to move molecules to the nucleus of cells. It's used on the bench as a gene therapy tool. It's actually found in some of the plasmids that are made by biotech companies for this exact purpose. Like if you're on the bench and you want to get something to the nucleus of a cell, you use a plasmid with this SV40 promoter enhancer, and boom, boom, boom. But the question is, first of all, why is it like what the hell is it doing in as part of the plasmid that was used for production of these COVID shots? Because it doesn't need to be there. There's already a promoter to induce the in vitro transcription reaction called a T7 promoter. Never mind. But the bottom line is it doesn't need to be there as part of the methodology. And the fact that it's a functional molecule and the regulatory bodies know that it's in there and have claimed that it's not functional, which is a lie, really makes you wonder what the hell not only it's doing in there, but what is it doing to people? Because, and this is the important part, it is a foreign DNA fragment, but it also binds P53, this guardian of the genome. It's a tumor suppressor gene, which is essential for tumor prevention. It does all sorts of other crazy things. If you ever speak to Kevin McKernan, he knows the details on this, but it basically throws a monkey wrench into the entire system and distracts, for lack of a better word, this very, very, very important tumor suppressor mechanism. And it doesn't take, it boggles the mind that we brought this to the regulators' attention and that there wasn't an immediate investigation. Because A, it wasn't disclosed, which is fraud, and B, hello, what about the physiological implications of all the people who are injected with the Pfizer products? We don't find the SV40 in the Moderna products, by the way, which means that it doesn't need to be there in order for the genetic material to be translated into spike protein or as part of the manufacturing process. But it's absolutely, like, there's no way that it's not having a negative effect clinically. And I dare say that it's in the context of cancer. Like all of these weird ass cancers that people are reporting, I'm absolutely sure those people had the Pfizer shot. And
Yeah,
it's related to this SV40.
Yeah, that'd be an interesting study comparing Moderna to Pfizer on that. And guys, just let me tell you, SV, simian virus, that's monkey virus. Old school vaccines like the polio vaccine had to be grown in some sort of cell, live cell. They use these kidney monkey cells to make that polio virus or vaccine. And so in that production process, it picked up some of this viral material from this monkey virus, simian virus 40. Well known, lots of studies and books published on this topic from years and decades ago, that it increases cancer rates, it promotes cancer, and it suppresses, like Jessica just said, the P53 is a cancer suppressor gene, and that simian virus 40 suppresses that. So there's just multiple ways that this can promote cancer, and that is what Kevin McKernan found in these vials. And that's just one thing.
Well, we found, me and Kevin and Charles Rixey, we did 2 DNA quantitation assays yesterday and sequencing, and 8 brand new vials, and we found it again. We haven't published this yet, but we will. But yeah, it's just it's not a question of whether or not it's there now. It's how much in each vial. And the fact that we kind of have to do this, under wraps, and it's hard to get our hands on vials. It's atrocious. This should be a worldwide effort right now because the world is affected.
Yeah.
Not even by choice. Most people were coerced into this crap, and now they're suffering the consequences of an experimental product that didn't seem to have any GMP practices going on.
Yeah. So besides just the toxicity of the actual spike protein itself, the toxicity of the contaminants, plural, we've talked about a number of them here, and there's probably more. The fact that it can be incorporated into the DNA. Is no wonder we see such a huge adverse event reporting spike and deaths spike. Jessica, talk a little bit about, this would well, before we go there, are there other things from a research standpoint that you're looking at, or is it just continuing to confirm what's already been found, just to bring more and more overwhelming evidence until the regulators would finally do something?
Well, I don't think the regulators are going to do anything. I mean, their job now is to continue to lie. I'm sorry. It's just come to a point now for me where you can't look away from the obviousness of the truth. They're lying at this point. What I witnessed in the last 2 days, because I did this work myself. I was the one doing the pipetting. I was the one prepping the samples. I was the one watching, the vials, get their seals removed. There was absolutely nothing wrong with our methodology. I did this myself 3 different ways. I think I'm still kind of shocked by it, because it's one thing to report what somebody you trust did. It's quite another to do it yourself and confirm it. And the horror is that, and the reason why I think the regulators will just keep trying to cover their tracks, is because it literally took us a few hours and just a little, small bits of equipment, which these manufacturers and even the regulators have in droves. It took us a few hours and a couple of reagents and just a little bit of man energy, or woman energy, or whatever you want to call it, to do this. It didn't take any effort, time, or money. So, if they cared about this issue, it wouldn't have become an issue in the first place. You see what I'm saying?
Mm-hmm.
They have the resources. They have more money than brains, to have been doing the proper tests back in the day before anyone got subjected to this. But of course, any results that they knew might have revealed what they were doing, which is why they cherry picked, which quantitation methodologies they were going to use for DNA and RNA. Um, they yeah, they didn't have any interest in doing due diligence in terms of maintaining some kind of standard. And that means like quality standard of product and health standard as per like health policy. The whole thing it's insane. If people actually knew how simple, the problems they've created are, at the same time they've created this insanely complex situation, people would be shocked. It had to stop at the regulators. Did you watch the movie The Big Short?
Mm-hmm.
So you
Yep.
remember when the people who are supposed to judge whether things were AAA or B or whatever, like the value of these bonds were, if they had actually not sold out, none of that would have happened. Do you see what I'm saying? They were the stopgap. If they'd said, of course we can't do this because they'll just go next door to the next the next agency to get the rating that they want. If they hadn't done that, if they'd actually been principled and not committed fraud and done their job, that whole thing probably wouldn't have happened. And by whole thing, I mean, the collapse of the housing market.
Yeah.
It's the same thing. It's like there's just fraud written into the code of how we're living right now. It's sad. It's pathetic. Which is why I think, I'm stressing now for at CHD. I mean, I literally probably talked to almost 1,000 people because people were just coming up to me. And if I gave any piece of advice, which I try not to do, but if someone would ask, I would say, maintain your sovereignty, be self-responsible in every single way that you possibly can. Coming up against this and realizing the deplorability of the truth, because we have, we as a public kind of have had faith in these agencies for a long time, probably most of our lives, which is also why a lot of people, they're still protecting themselves from engaging in the truth that it's possible that these agencies are just, they're driven by profit. They're not driven by the impetus to maintain public health like I said, I don't believe in public health anymore.
Yeah. Yep. I think that's probably the hardest thing for folks to, especially maybe the older generation. The younger generation probably more open to this thought, but it's a very difficult mental hurdle for people to get over, and many don't. They come to that hurdle and they turn around and walk back. They can't do it. To think that these agencies could be that corrupted to the tune of millions of adverse events and deaths. I mean, we've had Ed Dowd on here. It's been a few years also, guys, if you don't remember that. Cause Unknown is the name of his book. He was an insurance actuator and worked for BlackRock, financial guy. But anyways, he started studying the data on these excess deaths early on. And so this is old news, but new news as well, as Jessica just said, coming out of the lab just yesterday with this data. Amazing. But I know it's heavy. It's hard. I will say probably the most impressive thing I think at CHD, Jessica, after the kind of heavy stuff you presented and some of the other presenters too, but yet you found an ability to have some joy and happiness. And I saw you cutting a rug and dancing a little bit there and smiling. So there's still a way to find joy despite what can seem like a pretty kind of a desperate situation. But so maybe, I don't know if you have anything on the positive side to say, and you just did. I mean, take self-responsibility, guys. Get informed, get knowledgeable about stuff, take care of your terrain, you can decide what you're going to eat, how you're going to move today, how much sunshine you're going to get today, how nutrient dense your food is, how anti-inflammatory your food is versus pro-inflammatory. Despite what all this awfulness that's in these vials, the human body's incredibly designed. It will try and try and try and try to defend itself. It's got all kinds of backup and second and third options to go to. So if you just steward that thing, it can overcome. It's amazing.
Yeah,
Um,
totally. It's all about balance. So
mm,
you have to do like self experiments and determine like what balance is for you, because not everyone's the same too, um, and not everyone's being affected by these shots. Even the people who weren't injected, from shedding, we're not all being affected the same way. So yeah, you have to pay attention, and um, you have to be selfish in the best way. You have to be. You have to take care of yourself, because if you're a rack, you're not really, and you're not the best you. It might sound harsh, but you're kind of useless to everyone else. So it kind of plays on this predatory notion that they use, that you have to do this for the greater good. I mean, it seemed like everyone fell for that, but like, if you want to do the greater good, you got to go inside. Like, you have to go inward and figure out what's best for you. Optimize mind, body, soul.
yeah,
Yeah, there's only one you.
that's
um
right.
it's a harsh reality for most people. We come in here alone and we go out alone, and but it doesn't have to be sad, even if it feels harsh. It's kind of wondrous. And I think people need to like tap into that wondrousness and the magic that it is to be alive.
Yeah, I agree. And animals are a great way to tap into that, just like your beautiful
And
cat
it's
there.
with their butts right in front of your camera when you're in the middle of an interview.
Oh, that's great. All right, Jessica. I don't know what's the best way for people to follow your work when you do publish this study that's coming out, or just in general follow you and your education that you put out there.
Well, I just published a new Substack, jessicahar.substack.com. I'm also a big tweeter, I suppose, even though I'm highly suppressed on Twitter or X or whatever. Um, yeah, my website's still down, and I haven't really had time to get to that. But one of these days, I'll have a nice website. And yeah, just if you want to pop my surname into PubMed, you'll probably find my latest publications. Even though it's very hard to get any work relating to these shots published or through the peer review process, we are succeeding. So yeah, you can check out some of my work there.
Awesome. Well, Jessica, thank you so much for your hard work, you and your colleagues, but to do this deep scientific stuff that the clinicians rely on researchers like you. So thank you. And thank you for being a vocal voice for the general public to be able to turn to a trusted source to get some knowledge and information so they can make the best decisions for their families. So thank you for joining us kind of short notice after CHD and in the middle of your busyness. Appreciate you. We'll have to have you back in the future.
Anytime.
Okay, guys. So that was a pretty intense interview, kind of depressing, pretty heavy, but keep your chin up. There's always hope, and the hope's not in me or some supplement, although there are some supplements. If you've got the spike protein in you, we've had some success, and other physicians have had some success. You can go back and listen to that interview I did with Summer Powers. But we've seen Dr. Peter McCullough's Ultimate Spike Detox from the Wellness Company, plus Foreign Protein Cleanse. That's from Dr. Brian Artis. Ivermectin, nicotine, using this combination of things, using a binder. So there's some protocols out there that are showing some good success. So that Revasca supplement we talked about a few weeks ago, with Hans Vink and Bob Long that heals the glycocalyx. A lot of this damage from the COVID spike protein is the damage to the glycocalyx and the vasculature. So there are a lot of things you can do. But I want to encourage you that I believe, personal opinion, the most powerful thing you can do is get your heart beliefs right. Get lined up with righteously aligned with the truth, who is a person. Holy Spirit will lead you into all truth. That's the ultimate answer. So don't take this frustration or anger or bitterness or whatever emotion you have and put it into some scheme, trying to figure this out in your own mind and your own ways of doing things. Seek first the kingdom. That is it. We've got to personally look within and just the simple steps. And number one, believe God is who he says he is. We've got to clear up that. If we don't have a firm belief and understanding of God's character and nature, that's number one. And then number two, put off the old, put on the new. We got to renew our mind. And a lot of that's on our own stuff, what we believe about ourselves, these false identities. Go back and listen to Jamie Winship, because really, I frame everything almost now that I come across, including this interview. I'm going to frame this kingdom versus empire. And if you don't know what I'm talking about, go listen to the last interview I did with Jamie Winship. I've done 3, so it'll be the third one. Just put his name in the search bar on our podcast archive page on our website. Kingdom versus empire. That's it. So what are we going to do about this? Well, number one, we've got to believe God, believe who God says we are, walk in that true identity, put off that old false identity, understand who you are as a son. That's neither male nor female. Go back and listen to Leif Hetland's interview. Listen to Robin Perry Braun about sonship. We got to take captive our thoughts. We got to believe the truth. The power of our thoughts are just unbelievably powerful. Go listen to Carrie Kirkwood, The Power of Imagination. Andrew Womack, same topic. He has a book on that too. I've interviewed both those guys. Steve McVeigh, Quantum Life. I think 2 interviews with him. But all these guys are talking about the same thing, the power that we have. As a man thinketh in his heart, so is he. The power of life and death in the tongue. We do not realize the kind of power that we have that can overcome these schemes of the enemy, overcome spike protein, overcome DNA contamination. The one that's in you is greater than the one that's in the world. We have an incredible amount of healing power within us because of what Jesus did, what he bought and paid for, and what he gave us access to. But we've got to understand it. God's word is absolutely true. It's not just some Bible verse. So be encouraged, guys. That's the reason I interview so many of these kingdom guys like Chris Blackaby. Go back and listen to his interview. Just powerful. We got to see ourselves seated in heavenly places in that third realm. Same with Chad Gonzalez. His was exactly the same point. Our identity seated in heavenly places, and what that does to our physical cells when we can believe this stuff. The identity stuff is probably the most powerful. Ray Sturdivant, go back and listen to that interview, his book, Beating the Devil with His Own Stick. A powerful interview on identity, false versus true identity. Of course, the Jamie Winship series too. Jim Richards, Abraham John. So go fill your mind and then your heart with truth. Holy Spirit's got to write that truth on your heart. So ultimately, you need to have a conversation with Holy Spirit. That's the counselor. That is the truth. Holy Spirit will write the truth that you need on your heart. And then corporately, as the body, that's how we get something done. Not in our own strength, not in a political thing, not in anything but a kingdom of God. That's how we're going to overcome this thing. Jesus has overcome it. And now as his body, we're going to see these things manifest in the earth. It's happening, guys. So just be encouraged. Keep walking on your personal spiritual journey of maturation so that you won't be deceived and won't be knocked to and fro by every wind and doctrine. Okay, guys, we'll be back next week with another great show. Love y'all. Bye-bye.